Okay. All right, we've already got people lining up. I like it. So again, those of you online, we have been collecting your questions. If you've got more, go ahead and type them in now. We'll be mixing them in with the questions from the room. But let's start with the questions here in the room. At microphone number 1, step up nice and close for me, please.
Hi, thank you for the presentation. So I have learned a lot from the research the agency did on the LPLDA. Actually, I was in that field for many years. So I'm just wondering if you didn't address for the degradation profiles, how the agency will recommend to characterize the PLDA's degradation profile for drug product.
Yeah, I think degradation profile is definitely a very important component during formulation development, and often, that is one essential piece of information we look at when we're trying to really understand the release mechanism of a product. So here, just want to clarify. So you are trying to ask in terms of how we view like the importance of the degradation profile, or if that has been taken into consideration when we use that to support, generic.
Yeah, that's for the if we have the drug product ready for files. So typically, I haven't seen any guidelines how you characterize the degradation profiles. From my experience, I noticed that when you incorporate the drug into the polymer solutions, when you produce the implant, typically I did the side the comparisons. Typically, you see the difference from the the placebo group without the drug with the drug group. Typically, they didn't show exactly the same profiles, which is really hard to explain. So from a research perspective, but for the filing perspective, I don't know how the agency will require under what's the guideline you guys will develop for the industry to take the advantage reference the use standards.
Sure. I mean, as far as I know, I don't think we really have developed a clear guideline as for how to, like in terms of the key components should be included in your application. I think degradation profile, if you have done some research on that, that definitely would be helpful for the reviewer to better understand your product. But I don't think we have very well defined in terms of, if that's an essential piece. But that depends to, depend on your application and purpose. But like you said, the degradation profile is definitely very important.
Yeah, exactly.
And then, having that piece of information in the application will be helpful, for us to assess the overall drug product.
Yeah, but in that case, so if
Bing, did you have something you wanted to add there? Go ahead.
I can add something on to Yan's talk. So this morning we talk about lots of our PANDA program or control correspondence. Sounds like you have a perfect reason to submit a PANDA to the agency, so we can discuss your concerns, including those filing requirements, since we don't have
the guidance. Industry. But my point is, if we...
And that point is going to be a wonderful discussion during the networking event later this afternoon. Right. It's a good thing to have down at the
happy hour, so we can...
Okay,
Let me go ahead and move over here to microphone number 2, please.
Oh, thank you for your presentation, and very helpful. And I do have a question about the in vitro release. I saw you have the publication about the in vitro release study for the bupivacaine liposome. So I was wondering if you're going to propose a guidance based on this finding.
I mean, for now, that is one of the remaining scientific gaps we have identified when we develop the guidance. So, in general, if we are very confident, or comfortable with one guidance, we would include that in the FDA dissolution database. For now, I'm not sure if we're there yet, but that's something we are continuously working on and trying to eventually come up with a method or through the publication to provide more guidance on the method development for this dosage form.
So right now you don't have a.
Yeah, so then I think that's a good point in terms of, when you review or read our publications, you still should treat that the same as all other literature references. It doesn't, represent FDA's policies or review standards.
Okay. Yeah, because it's sometimes it's very challenging for these kind of complex drugs. If I have in vitro release, like especially like for that some in situ forming product. Do you have any plan on that?
Again, wonderful question to continue at networking downstairs. For these where we're just going to keep digging until we get the answer we want, we can do that at the networking event. Can I get an online question, please?
For controlled correspondence for a Q1, Q2 determination for liposomal product, what additional information other than the proposed formulation would be needed for this determination? Any characterization data needed?
Just to clarify, did you say for liposomes?
Yes, liposomal product.
Okay, I think that question is out of the scope of current session.
Okay, then let's move over to microphone number 1, if we could, please.
Yeah, hi. I have a question about your discussion of safe spaces and how it would relate to setting acceptance criteria. So you mentioned that if you can demonstrate sort of a worst case on the upper and lower range of a profile, anything within that could be deemed bioequivalent. So for setting that for a reference product, is there, or do you anticipate any limits on the width of that? Say we see very wide worst case situations, creating a very wide target. How do you anticipate that's going to be addressed?
I think if you can provide the data supporting that safe space, we would definitely review that.
Thank you.
All right, microphone number 2, please.
Continuing on the same thought, I noticed the last presentation was in vitro release testing for long-acting drug products. I know you had a few examples in there for long-acting, but it seemed like maybe it was applicable to any in vitro release method. Is that right?
Extent?
Okay, thank you.
All right, microphone number 1, please.
I was wondering if you could just comment generally on the expectations when you have an IVRT, in vitro release test, used for quality versus when it might be used to establish bioequivalence. Do you have any sort of general thoughts on any differences between those two applications?
Hello. So I think whatever I talked was from the product quality perspective. So we focus on your proposed product when we are looking through the quality perspective. I think my colleague probably can talk about the bioequivalence aspect.
In general, in terms of method development and validation, the general expectations for that component is the same. But then when an IVRT used as a part of the BE approach, then it's not necessarily, you can have a different method, from the one you will use for the QC, because this will be a one-time study to support the BE assessment.
Okay, online question, please.
We have a related question. Industry is getting confused on in vitro release method and dissolution method. I see the in vitro methods from the bioequivalence guidance runs longer, such as many days, and practically not possible to run the same method in QC for many days to release the product. Does the agency agree that the dissolution method can be different than the in vitro release method, and can it run in a few hours?
I'm not sure if I exactly understand the question, but I think the question was more about a long in vitro release method versus a short-term in vitro release method from regulatory QC perspective. So I think as I mentioned in my presentation, we do understand that when a long-term method cannot be used due to practical limitations, you can explore developing an accelerated method that can be used as a regulatory tool, as long as you provide information based on the cross-validation, interchangeability of these two, and provide us some data to show that both the methods have equal, or the accelerated method has more discriminating ability than the long-term method.
Yep, please.
So I want to add around the previous questions. I think it's very good questions. I want to expand this a little bit, not just in vitro release testing. This also can apply to other quality tests, right? So there's twofold. The first fold, you establish a bioequivalence using in vitro approach. I think the question from some Q3 attributes. So when this translates into quality controls, we may be talking about a different sets of quality controls. Like you mentioned, the release testing, if you have long-acting drug, 12 months or 12 hours, 24 hours, it will have 7 days transdermal patches. We're certainly not going to ask you to do a 7 days dissolution test. Same thing as adhesive rheologic test. So from quality perspective, we ask you have different sets of control, but the critical thing or the very important thing is you have to establish some kind of bridging study to demonstrate how the in vitro test you used for bioequivalence or in vivo bioequivalence transfer to the quality controls.
Very good. Microphone number 2, please.
Hi, great presentation. Thank you all. I have a question regarding how to identify the critical material attributes. So for the container closure systems of complex products, there's some parts that is not in direct contact with the formulation nor with the patient usage. Is the DMF required for such product and materials?
Again, this sounds like not related to those wonderful talk we had in the last sessions, since I'm kind of outsider, but maybe I can address your questions. So you're asking a complex product, maybe have a container closure, has no impact about the drug product. I just randomly give, say auto-injector. So you have a cartridge in there, you have a device. Actually, tomorrow morning we have another session, we'll talk about that. But I give you some just heads up now. It depends, right? You have to demonstrate the secondary packaging system has no impact on a primary container closure. Maybe it's okay, but maybe not. So again, you maybe can submit either control, depends on the complexity. You can either submit a control or you can submit a request for pre-end meetings. But we're going to talk about more device related issues tomorrow morning.
Okay. Microphone number 1, please.
Hi. My question is related to low soluble API, for which you explained that sometimes you don't even reach 80% of the release profile. So for those products where the method is very difficult to develop and you don't even get 80% release during the release time, at stability it is further very slow. Can we have 2 different methods, one for the release and one for the stability, with difference in some enzymes to to help the solubility?
I think we generally recommend same method to be used for release and stability. I do not get the part of adding the enzymes in here because sometimes it's more focused on the capsules, and we understand that limitation for that particular dosage form. But we generally recommend identical method to be used for both release and stability.
Thank you.
Online question, please.
How many lots of drug product should be characterized for polymers PLGA And should characterization include raw material PLGA characterization?
Usually recommend categorize at least 3 lots of drug product.
Alrighty then. Microphone number 2.
Thank you for the presentation. So for the polymer containing formulation, when we send control correspondence for Q1, Q2 sameness evaluation, shall we include the characterization data in the control correspondence? For example, the molecular weight or the L2G, like the ratio?
You are asking when you submit a control correspondence for Q1, Q2 evaluation of, for example, PLGA, containing PLGA component, whether you need to submit the categorization data at the time. Yes, when you submit those control correspondence, you need to provide those additional categorization data to help us to evaluate whether your formulation is Q1 sameness or not. And then we can further determine whether it's Q1 or Q2 sameness.
Yeah, and just
Go ahead. Oh, yeah, please.
just to add on top of that, when you submit the characterization data, it's really important to have the characterization data on the finished product. If you feel like it can provide additional supportive information, you are welcome to provide the characterization data on the raw material as well. But when we assess the Q1 sameness, really we are looking at the finished product.
Microphone number 1.
In your presentation, you have given 5-6 tests for characterizing the PLGA. So does agency have any view to include those tests in the DMF of PLGA?
I'm sorry, I didn't quite catch the last part.
There are 5-6 tests which were given in your presentation, like, the LAGA ratio, molecular weight, crystallinity, TG, and all those things. But that is for the finished product. So do the agency also expect for the DMF, in the DMF also the manufacturer should include that data as a part of DMF, excipient DMF?
Yeah, I would think so. Bing can probably
Can you repeat the question real quick?
Oh, yeah, I think the question you're asking is the characteristics of the PLGA, whether those information should be included in the DMF of the polymer, right?
Yes.
Yeah, my general impression is those should be included to show, how the PLGA is consistently synthesized.
But the same polymer may not be used it may be used for a different product also. So how it will be correlated to the particular product.
So you mean like you are referencing a DMF which has already been used?
Yes.
Yeah, so I think that's okay. I think Bing can probably chime in.
Yes. I mean, you always can option, even we have type 3 DMF. So if you have something commercialized polymers, if you have access to a DMF, you can reference the DMF and we'll review the DMF. We'll review the information provided in DMF.
Thank you.
Online question, please, Ray.
Regarding glucose PLG polymers in the sandostatin LAR product, does characterization data indicate whether the glucose is present in the commercial product in a closed ring or open chain form?
So the question is asking if we have done any study to verify the structure is like a star shape or is more close.
Maybe let me take a stab at this question. The question is really want to ask that since glucose can exist in an equilibrium in a chain form or in a ring form, and here it's asking whether in the study have data indicate which kind of form the glucose might be.
Yeah, in the present study, we don't have any information about the form of the glucose.
Okay, microphone number 2, please.
Thank you very much. All fascinating presentations. Now, I feel there's some submission because you talk about the molecular weight, talk about LG ratio, talk about branching unit for excipients. But we know that the microspheres themselves sometimes are as important as excipients itself. So, for example, particle size, surface area, particle porosity, and even small amount of impurity could catalyze the degradation of microspheres. So that's why I feel there's something missing. So do you have any comment on that?
Yeah, actually, the scope of the study is try to develop a method and tries to fill the scientific gap on the area of how to characterize the excipient itself to demonstrate Q1 or Q2 sameness. But as you mentioned, some other quality attribute in the whole product, for example, the particle size or microsphere and the porosity and some other characteristics, they are overall will play a critical role in the drug release. Yeah, that's different scope of the question.
Yeah, I mean, your point is valid, and we do agree all those things of the finished product is equally, if not more important than the characteristics of the raw materials. It just happened to be for today's presentation, we highlighted and focused on the studies on characterization of the raw material. But besides those studies, we actually have also many other projects focusing on the aspects you just mentioned. And we do have a few publications already on just to study the formulation characteristics such as porosity, and then also other things, how those are affected by manufacturing raw material changes and how we can properly characterize those.
Thank you.
Okay, microphone number 1, please.
Thank you very much for very good presentations. In your last presentation, you mentioned developing a clinical relevant IVR method and establishing a safe space for the specification. And you mentioned that it could be through IVIVC development or IVIVR. So beside IVIVR, do you expect to do the modeling approach?
I think it's up to what data you have, and it's up to you to decide. So it can be based on rank order, it can be based on a pilot study and a pivotal study, and of course it can be based on the PBPK type of modeling approach. So it depends on what data you have.
So it could be either IVIVC, IVIVR, or PB modeling.
Yes, and each of those, as you would know, have their own advantages and limitations.
Yes.
Okay. Online question, please.
For a long-acting complex injectable that the excipients are slightly different, can we submit a meeting request as opposed to a Q1, Q2 proposed formulation controlled correspondence?
I think the question is asking if they tend to use an excipient which is slightly different from the ones used in the reference product, instead of directly submitting a Q1, Q2 control correspondence, whether they can send a pre-NM meeting request. So I think this is, I think the short answer is depends. So if, that depends how much information and homework you have done. If you have done enough, studies to really support why, this difference should be allowed and are not expected to have any negative impact on product performance, then you have specific questions for us to address, and you think it's beneficial to have a, direct discussion. Then we encourage you to, have a pre-NM meeting request. But we can't really have a simple answer to your question without seeing the actual content.
All righty. Microphone number 1, please.
My question is for Quinn. You mentioned in your presentation blockiness of PLGA is critical. Do you have any recommendation for the methods to be used? There's no specific guidance given by FDA on that.
For the blockiness of PLJ, actually, Akina, they already published the paper reporting some of the results using the carbon NNR using NNR method to characterize the blockiness of PLJ. You can look at some of the publications.
All right. Yes, microphone number 1.
Yeah, in your presentation, especially the dissolution, where you have mentioned like, IVFS is like probably a good idea to, going forward, if any change we do in manufacturing can be useful. But considering the nature of complex injectable and it's long acting, most of the studies are in patient, and having 3 different formulations, high release, low release, and target release will be a challenge. In that case,
they should use. So based on our current research, the GPC coupled with 4 detectors using dynamic light scattering, all those detectors together by measuring the inherent intrinsic viscosity of the glucose star polymer appears to be one of the most direct measurement of the branching frequency. That's all we can see for now.
Okay, let's go ahead and take, no, let's not take one more online question. Even online is saying, okay, that's enough. All right, so before I go into the wrap up for the end of the day, please help me thank our panel for coming and talking to us.